Good morning, students. So today we are going to discuss about nucleus. So nucleus is one of the prominent structures found in the cell, and it is called as the brain of the cell. Why? Because it controls all the activities of the cell, and it has the basic nature. It determines the basic nature of the cell. Nucleus is called as the control center of the cell. Nucleus contains DNA. Nucleus is surrounded by a double membrane, and it is usually the easiest organelles to see under a microscope. And nucleus, usually one per cell will have one nucleus. And remember, the cell which have the well-defined nucleus and the nuclear membrane are called eukaryotic cells, found in higher plants and animals, whereas the cells which does not have the well-defined nucleus and the nuclear membrane are called the prokaryotic cells, the primitive cell found in the bacteria and other minute organism. So let us see the nucleus. See here the nucleus. The various structures are there. Nuclear envelope or nuclear membrane. Nuclear pores are there. Then we have the nucleolus in the center, chromosome in the inside the nucleus, and chromatin, the reduced form of the chromosome. So let us proceed to the chapter that is given in a page number 110. Nucleus, the brain of the cell. So nucleus is called as the brain of the cell. In the cytoplasm, almost all cells, there is a prominent structure called the nucleus. Nucleus is often referred as the brain of the cell because it determines the basic nature of the cell. The nucleus is bounded by a double membrane called the nuclear membrane or nuclear envelope, lined by an inner layer called nuclear lamina. A jelly-like nucleoplasm is there in the nucleus, and in the nucleus lie a tuft of twin structures called nuclear reticulum or chromatin. And inside the nucleus plasm we have a dense structure called nucleolus. So nucleolus helps in the synthesis of ribosomes. So nucleolus helps in the synthesis of ribosome. And chromatin materials are this form of chromosome. When cell division starts, this elongates to form the chromosomes, which carries the genetic material. So during cell division, the nuclear reticulum or chromatin opens up to form the distinct thread-like structures called chromosome. Genes that determine all the characteristics of organism reside in this chromosome. So remember, genes are present in the chromosome, and chromosome is present in the nucleus, in the nucleoplasm. Do you know plasma membrane is formed of protein and fat molecules and is actually a fluid. Now come to the structure of cell nucleus. So cell nucleus look like this. So nucleus contains the double membrane nuclear envelope or nuclear membrane. Inside we have the inner lining or nuclear lamina. Then inside the nucleus we have the nucleoplasm, the jelly-like substance. Inside the nucleoplasm we have the chromatin or the nuclear reticulum. And this chromatin becomes thread-like structures during the cell division, which is called as chromosome, and chromosomes carry genes. And in the center we have the nucleolus, which helps in the formation of ribosome. Here the ribosomes are there, the granule-like structures, and we have also the nuclear pore. So this is the structure of cell nucleus, and the function you all know that it controls the activities of the cell. So it is called as the control center of the cell, and also it carries the genes through the chromosome during cell division. This is the main function of nucleus. Now come to the next page. Here it is an activity is given plant cells. So what you will do, you will take an onion bulb, slide covered slips, two watch glasses, a needle, a brush, a compound microscope, a blotting paper, a methylene blue solution, glycerine, and some water. Procedure: take an onion, slice into half, using forceps, pull out a thin membranous peel and put this peel in a watch glass. So take a small portion of the peel in the watch glass containing water. Mix 1 or 2 drops of methylene blue so that color will stain the bluish color. So leave the peel for about 3 minutes, then again wash with water so that extra stain should be washed away. Now take a clean slide. Slide means of glass. Put a drop of glycerine in the middle and slowly put with the help of brush the stained peel, and then carefully place a cover sleeve, a small square glass piece, very slowly, lowering it with the help of needle. And while doing so, make sure that there should be no air bubbles. If air bubbles will be there, then it will be difficult to see the compartment of cells. So remove excess glycerine from the edges of the cover slip with the help of blotting paper, and then observe the slide under the microscope. So you will see the first in the low power and then in high power. So in the low power you will see the compartment, where in high power you will see the dot-like structure. So what is this dot-like structure? The nucleus. So you will observe that small compartment-like structures are called cell, and you will also notice a small dot in between the compartment that is called nucleus. So this is the nucleus, the dot-like structures here, and all cells have the distinct but similar shape. The shape of each cell is given by an outer membrane, making them look like compartment. So make a diagram of what you observe under the microscope and note down in your laboratory notebooks. Observation, discussion. The small compartment that you see are actually the cells. However, the cells that Robert Hooke observed are very different from the cells you see here. So these are the living cells with materials inside, while he saw cells that were dead and empty. There was no dot, no nucleus. So Robert Hooke saw the cell, the dead cells, and that was empty, that does not contain the nucleus. So living cells contain the nucleus. So that is about the nucleus. So let us see the checkpoint. Match the following organelles. Plasma membrane. So plasma membrane, that means control what leaves and enter. So this will be the for plasma membrane. Nucleus, brain of the cell, 1. Mitochondria, powerhouse, 2nd. Then the Golgi body, that is Golgi body have the function transport within the cells, endoplasmic synthesis, packaging and storing the cellular materials, ribosome synthesis protein, and the lysosome destroys worn out tissues. In this way you will write the checkpoint. So that's all for today.